Chromosomes clash day and night. Genetics laid bare under light. Bunsen burners flicker bright. All in the name of Mr. Right. Doing science right tonight. Answers never out of sight. Biology battles fierce and bright. Learning hard and feeling right.
Hi Year 10s, and welcome to your first trimester of biology. So this is the 10-week course of biology that we're covering at Saints. This is the very first PowerPoint. What is DNA? This is the first topic of DNA and genetics. So focusing on this first PowerPoint of what is DNA, at the end of this PowerPoint, or the end of this week that we're looking at this in class, you should be able to tell me that nucleic acids, DNA and RNA are made up of nucleotides. You should be able to tell me what are the differences between DNA and RNA. You should be able to tell me that DNA is a double helix made of two antiparallel strands, and also be able to tell me about the hydrogen bonding between those strands of DNA and the complementary base pairings. You should be able to, for the 2 dot points down below, talk about the 3D models that Watson and Crick were using when they discovered the structure of DNA, and you yourself need to be able to draw DNA and RNA strands and nucleotides. All that's great. Let's go into some content. What is DNA? For almost all living things, DNA, or deoxyribonucleic acid, that's the molecule that provides instructions for the organism's characteristics. Essentially what it is the DNA codes for proteins. Your body makes those proteins, and those proteins act as enzymes and other sources to go along and do chemical reactions in your body to ensure that the characteristics that you have represent you. DNA is really useful in terms of helping provide diversity amongst living things. Slight variations in DNA gives us those different appearances. The actual structure of DNA was discovered in 1953 by James Watson and Francis Crick. DNA was discovered before that, but the structure of DNA was discovered in 1953 by James Watson and Francis Crick, and that model is known as the Watson and Crick model, which you'll get to know quite well throughout this topic. So, nucleotides. DNA and RNA are both made up of what we call nucleotides. Nucleotides are the building blocks of those molecules. In every living organism that contains DNA, which is every organism on Earth, DNA has the exact same structure. Always made up of repeating molecules of nucleotides. And each nucleotide is always made up of the same 3 things. We're starting off with they're made of the pentose sugar, or deoxyribose sugar, as we call it, which we draw as a pentagon. Okay. From that, we also have a phosphate group. Now, the phosphate group is always attached to the 4th point on the pentagon. So, starting as this number 1, 1, 2, 3, 4. From that 4th point, we attach a circular phosphate group. Okay, and every nucleotide also contains a nitrogenous base, which is connected to the 1st point in the pentagon. The 1st point in the pentagon starting here, we draw our rectangle for our nitrogenous bases. Okay, now, those nitrogenous bases are usually one of 4 bases. So it could be adenine, thymine, guanine, or cytosine. But we just call them nitrogenous bases, and then all nucleotides have those same 3 components. This is one nucleotide. If we were going to combine multiple nucleotides of DNA together to make a strand of DNA or a molecule of DNA, then you would attach this phosphate group here to another pentose sugar, and that pentose sugar, or that phosphate would connect to the 3rd point on the pentose sugar, so 1, 2, 3, there'd be another phosphate group down here connecting to another nucleotide, which you can see in this slide here a little bit. You can see the one, this one's slightly different, but you can see, sorry, that should have been the fourth point. Over here, we've got our phosphate group coming off on our 4th point. Then over here on our 1, 2, 3rd point, we're connected to the phosphate group of the below nucleotide. And our 1, 2, 3 point connected to the phosphate of that nucleotide. So the phosphates are always connecting to the 4th point of their nucleotide, and then to the 3rd point of the pentagon of another nucleotide. When we have these together, and we have our strand of DNA, which would just be one side of these, they come together and they form a double strand of DNA. So we can see 2 strands here. One strand is going down this way, and the other strand is going up this way. Okay. They then twist together, and it ends up looking like a twisted ladder, which we call a double helix. So this is the double helix model. Okay. Nitrogenous bases making up the rungs of the ladder, and the phosphate and sugar making up the sides of the ladder. Okay. You're going to be asked more to draw the nucleotides themselves, like we had in this image here. You're going to be much more likely to have to draw this, but you may be asked to describe why it's a double helix structure. When it's just we have those two strands of DNA, nitrogenous bases connected with hydrogen bonds, and then the sugar and phosphate sides of the ladder having a double helix shape. Let's look at those nitrogenous bases in a little bit more detail. So, like we said, in DNA, there's 4 bases that can be found. So we've got adenine, which we label with an A, thymine, which we label with a T, guanine, which we label with a G, and cytosine, which we label with a C. All 4 of those bases have very different structures, and hence they'll only bond in certain ways. So, for example, adenine will only ever bond with thymine, and we kind of say that with the straight letters will bond with the straight letters. Whereas guanine will always bond with cytosine and nothing else, G and C together. Curvy letters with curvy letters, G and C both are curvy letters. That is complementary base pairing. So if we go back and look over here, if we have a thymine on this side, then it has to be an adenine on this side. If we have a cytosine here, then it has to be a guanine on the other side, because we know that A and T bond together and G and C bond together. So you could be given one strand, and based on that, able to predict what is going to be on the other strand by knowing about that complementary base pairing. A little bit more depth into that when we're looking at this. The reason for that, if you're looking at those bonds, adenine and thymine are bonding with 2 hydrogen bonds. You can see the 2 hydrogen bonds between A and T, whereas cytosine and guanine, they bond by looking for 3 hydrogen bonds. As you can see in both of those examples there, they have 3 hydrogen bonds. Therefore, guanine can't bond with adenine because adenine's looking for 2 bonds and guanine's looking for 3. They don't bond together. So guanine's only going to bond with cytosine. Okay. So that's our nitrogenous base pairing, or a complementary base pairing. Okay. Talking about how DNA was discovered, it was discovered, the actual idea of DNA, in 1869. This was done by isolating an unknown chemical from the nucleus of a white blood cell. What they were actually doing is they were looking for proteins, and they found this substance that was markedly different from proteins in its chemical composition. It wasn't a protein, it had a really different structure. When that was discovered, they didn't actually know that they'd discovered the genetic code. They'd just discovered something unknown. It wasn't until 1943 that a scientist called Oswald Avery proposed that DNA was actually the genetic code in organisms, and that was then proved in 1952 by Hershey and Chase. So we're looking at 90 years later, or 80, 90 years later, we're actually, after discovering DNA, discovering what it's actually made up of, and what its actually role in the body is. Then we're talking about the DNA, the actual structure of DNA, and this was really competitive. Everyone wanted to be the first to describe what the structure of DNA was, and how it was used as this hereditary material. Lots of scientists involved: Rosalind Franklin, Watson and Crick. Lots of scientists involved. There's a few others. Wilkins is another one. And we'll watch a documentary on this in class. And they all had very different techniques of how they went about trying to discover the structure of DNA. Watson and Crick, their idea was to build models. They wanted to build 3D models and hopefully get a model that would fit all the properties of DNA, and then say, hey, we found the structure of DNA. They actually did this multiple times. They got some wrong in the first case. So their very first model, they actually made DNA as a triple helix with the bases all on the outside of the molecule, and then magnesium holding the strands together with iron bonds, because they had very little evidence, because they were just building models, and they thought, hey, this is exactly what it looks like. And then we get into Rosalind Franklin, who used X-ray crystallography to deduce DNA was a double helix, and it had phosphates and sugar on the outside and nitrogenous base on the inside. And she then, without showing them that information, said, hey, your triple helix is wrong. There's not enough magnesium for this to actually work, and therefore your model doesn't work. We then had Maurice Wilkins, who shared the X-ray crystallography that Rosalind Franklin had taken in the 1950s with Watson and Crick without her permission. They then used that information to make their own new updated model of DNA. which was correct. And that's the double helix model that we know today. The issue with all of that is Rosalind Franklin then died from radiation poisoning in 1958. Radiation poisoning from taking the X-rays of DNA structure that she technically had a major part in discovering, and hence never received the Nobel Prize for it, because you can't receive that once you've passed away. So only Watson, Crick and Wilkins received the Nobel Prize and the credit for discovering the DNA molecule in 1965, whereas Rosalind Franklin didn't really get any credit in that until 1968, a long time after Watson and Crick had actually published their data. So it's a little science as a human endeavour story, just talking about how DNA was discovered and the structure of DNA and the race to discover that structure. Like I said, we'll watch a documentary of that in class, and that will be a lot clearer. The last thing we're going to go through is the differences between DNA and RNA, like from that checklist. So RNA, this is another type of genetic information that's found in organisms. We actually believe that RNA was the first type of genetic information in early life. You'll learn a lot more about this in Stage 2 biology, Year 12 biology, because this is called the RNA world hypothesis and something that's discussed in the evolution topic. Main differences here, we can see that straight away by just looking at them. Unlike DNA, RNA is a nucleotide that is, or a genetic information that is single-stranded. DNA is double-stranded. RNA uses ribose sugar, which is the name ribonucleic acid. DNA uses deoxyribose sugar, deoxyribonucleic acid. So they have a different type of sugar. And lastly, with the bases, as we discussed previously in the PowerPoint, DNA is using adenine, guanine, cytosine, and thymine, but RNA can't physically make thymine. So it still has adenine, guanine, and cytosine, but it replaces thymine with uracil. So when you're dealing with RNA, if you have an adenine on the other side, then it will always put a uracil in place of thymine, because it can't make thymine. That's the end of this first PowerPoint. Hopefully this has helped you a little bit more with the content we've covered in class, and I'll see you in the next video.